Hello, my name is Vera Prytkova. I'm a research and application scientist at CCDC, and I'm going to briefly show you how you can work with the protein structure in Hermes. Take a look at Hermes interface. On the right here, you have a display window. On the left, you have a molecule explorer, contact management, a graphic objects explorer. In order to load the protein, you can click on File, Open. We're going to load the protein structure with the PDB ID 4IGS. This is a crystal structure of human aldose reductase. You can observe the structure in the display window on the right. By right-clicking the mouse, you can move your mouse and shift closer or farther away in the protein view. In the Molecule Explorer window, you can click on the arrow and observe what kind of structures are present. You can look at the number of chains, what are the amino acid names, what are the co-crystallized ligands, the cofactors, and water molecules. You can right-click on the waters or any other part of the structure, and you can remove it by clicking delete. You can change the appearance of the structure. Let's say we want to look at the ligand. We can right click on it and center and zoom 3D view. Now we're looking at our ligand. We can change the appearance of this ligand by right clicking on it, selecting styles and changing the style. This time we're going to change it to capped sticks. We can also protonate our structure by going to Edit, Add Hydrogens. We have 2,879 hydrogens added. We can save the structure by going to File, Save As. We can save it in the PDB file format or in multi file format or any of the picture formats. And we can also close our view by clicking on Close All Files. Thank you for your attention.